Good afternoon, everybody. I'm Donald Clark. I am one of the acupuncturists at CNY Healing Arts, CNY Fertility, and everything's been good this week. It's been the weather is starting to turn towards winter here, and it's getting colder, a lot colder, and it's about time. It's getting kind of late. We had our first frost freeze, which is good, kind of tamped down all those mosquitoes, and we had to say goodbye to the flowers. So today we have a special guest from Embryology. Maureen is here today joining us. She's going to kind of just go over everything that they do there and just give everyone just a little bit more insight as to the process that we do here at CNY Fertility. And I hope you enjoy it. You know, it's been a long time coming. I've been talking about it, and she's really stepped up, and she's brilliant. She's brilliant at what she does, and I hope you guys enjoy it. I enjoy talking to her. So we talked about, we've always talked about keeping your nest healthy, and in this process of fertility journeying, one of the big aspects of it, there's many, is follicular development, right? We want healthy, viable eggs, right? And I know you all have been so good at that, and you've been taking care of yourselves, and I hear about it, and I see the results, and I'm proud of everybody. So we're going to go a little deeper. We're going to go onto the western side of the of this process, and I hope you enjoy it. And text me, email me, set up consultations on Wednesdays, and I'd love to see you, or I'd love to talk to you. And I'll hand it over to Maureen.
Hi everyone, I'm Maureen. I work here in Syracuse in the Embryology lab. I was just going to go over a little bit of what we do in the lab, give you a little more insight into our processes and what goes on. Okay, so every day we start with quality control. That's the first thing we do when we get to the lab in the morning, is we check all the incubators to make sure they're at the right temperature. All of our workstations in the lab are at 37 degrees Celsius. We're trying to keep the embryos comfortable and mimic the human body as much as we can. All the incubators are hooked up to gas lines with carbon dioxide, oxygen, and nitrogen. So we check all the gas connections in the gas tanks. We check all of our frozen embryo storage, so all of our cryo tanks, to make sure they're looking good. And all of our incubators and cryo storage tanks are hooked up to a monitoring system. So we have an alarm system that will call us 24/7, day or night, and let us know if anything goes wrong with the incubators or the cryo tanks, and someone can come and check up on them. But we really don't get that many phone calls. So that's kind of how we start our day. And then we go right into our embryo checks. So we start checking for fertilization, check the embryo development. All of our dishes for the embryos we prepare the day before. They incubate in the incubators overnight so they can equilibrate and get to the right temperature and gas concentration. So I brought a little dish, a little culture dish with me to show you guys. Oh, where's the camera? Kind of bright. So you can see all those little circles in there are the different wells where we put the eggs and embryos. So we have 1 to 3 eggs or embryos per drop. And the drops are covered with oil, which again helps keep the temperature and the pH and the gas concentration the same for the embryos. This is my first live, so I'm going to be looking at the comments and kind of doing my spiel. The difference between a day 5 and day 6 embryo, we consider them basically about the same. We're just looking for an embryo to get to the blastocyst stage, which I will go through what that is in a little bit. But we're not really too worried about if an embryo gets to a blast at a day 5 or a day 6, and sometimes even day 7. So we'll freeze the embryo once we see that it's developed into a blastocyst, and some embryos do that on day 5 and some do it on day 6. If we're looking to transfer embryos that are not Pgta tested and you have day 5 and day 6 embryos, we'll generally start with the day 5 embryos because statistically they're a little bit more likely to be to have normal chromosomes, but plenty of day 6 embryos are normal and make beautiful babies, so we really don't worry about it too much. Okay. All right. So I also brought one of the little tiny pipettes that we use to move the embryos in the dish. So this is called a stripper tip, and it's like a tiny little plunger. It's super tiny, and we have these little plastic, they're called strippers, and it goes on the edge, which it's kind of dark over here, so give me a second. So it goes on like this, and this is how we pick the embryos up and put them down in our tiny little dish. So it's very thin. So I thought you might like to see that because I didn't actually see one of these until I worked in the IVF world. Okay. So have I seen a 2-cell and a 5-cell make good babies? I've seen 5 cells make babies. Yes. Yes. I don't know that I've seen a 2-cell on day 3, but it could happen. If the embryos are untested, besides being day 5, how do you decide which one is best to transfer? So we're just looking at the morphology of the embryo. So we're looking at anything that develops into a blastocyst, and it's kind of like a beauty contest, to be honest. Sometimes the best looking embryos may not work, and sometimes the one that we don't think look ideal will make gorgeous babies. So we're just doing our best to pick the ones that we think will work. If you have really good quality, good looking embryos, they are more likely to work, but that's not always the case. Sometimes the ones that we might pick second will work, or the ones we pick third. What's the difference between a day 5 3AA and day 6 6AA? They're pretty equivalent. So the AA means that the inner cell mass is good and the trophectoderm is good. So that's where the letter grades come from. So those are both excellent embryos. A 3 means that it's an expanded blastocyst, and a 6 means it's a fully hatched blastocyst. So it's already come out of its zona. It's come out of its shell, which I can show you in one of my pictures. If you held that day 5 3AA to day 6, it might fully hatch and be a 6AA. So they're pretty much equivalent. So I'll show you here, since we're getting into some embryo questions. So when we check the let's see. Okay, so day 1, you come in, you do your egg retrieval, we get the eggs. We are searching through the dishes from what the doctor extracts. We pick out all the eggs. About an hour later, we're going to strip all the outer cells off the outside of the egg so we can see the egg really clearly and see if it's mature. And then an hour after that is when we inject 1 sperm into each mature egg. And then the next morning, we're looking for fertilization. So I have a picture. This is a fertilized egg, and we're looking for 2 pronuclei. So you can see, it's backwards. You can see the 2 circles in the middle. So that's what we're looking for. This is a normally fertilized egg. It has 2 PNs. So that means 1 contains the DNA from the sperm and 1 contains the DNA from the egg. So that's what we're looking on day 1. The next time that we check the embryos is day 3, and we're either going to do a day 3 transfer, day 3 freeze, or we're holding the embryos to the blastocyst stage, which is days 5, 6, and 7, and we're either doing day 5 transfer, freezing the embryos, or doing Pgta testing. That's my Aunt Steph. Thank you, Aunt Steph. So here's an image of embryo development. So this is the fertilized egg. And then let's see, day 1, we're looking for 2 cells. Day 2, an ideal embryo has 4 cells. Day 3, an ideal embryo has 8 cells. Again, these are all ideal. So if you have a little more cells or a little less cells, that's fine. This is just in an ideal world. Day 4, we don't really check the embryos on day 4 because they start to compact. They become what we call a morula. So all the cells kind of compact together, and we really can't count them, and we can't really predict what the embryo is going to do from that point, and we can't really grade them on day 4. So they compact like this, and then we check them again on day 5. So then it becomes hundreds of cells from the morula stage to the blastocyst stage, and we're looking at the different parts of the embryo to grade the embryo. And then this is a hatching embryo, so it's starting to come out of that shell, which is why there's stuff coming out there. So the embryo has to hatch out of that zona in order to implant into the uterus. So I'll show you, let's see. This is a really good blastocyst embryo. So you can see the circular part here, group of cells, that's the inner cell mass. That's what becomes the baby. And then all these cells on the outside, looks like a soccer ball. That's the trophectoderm, and that's what becomes the placenta and the amniotic sac. So this is a really, really good embryo. This is like a beautiful AA, 4AA. So that's what we're looking at when we grade. So let's see. How do we pick the sperm? That's a fun question. So the andrology lab washes the sperm, puts them through the centrifuge, and gives us the process sample. And then we take just a tiny little bit in our stripper tip, we put it in our ICSI dish, and we're looking at the sample under extreme magnification. So like the size of the computer screen, they're just swimming around, and we can see the head, the mid piece, the tail. So we're looking for sperm, we're looking for like ideal morphology. So we're picking the best looking sperm with a normal shaped head, and we're looking for sperm to be progressive and modal. So we want them moving, and we want them moving straight. Sometimes you'll get all sorts of wonky looking sperm, and we try to pick the ones that look the best and are swimming strong and straight. Let's see. So I'm looking at this. You were told your embryos were frozen on day 2, but they were a 3-8 and a 2-6. Are they worth using for an FET? If you have a 3-8 and a 2-6, yeah, definitely they're worth using for an FET. Okay. All right, let me go back to my little chart here. Okay, so a lot of times we get asked why we do a day 3 sometimes or a day 5 sometimes. So that's going to be based on not only the embryos, but your age, your history, if you've done IVF cycles before, and what the doctor recommends for your treatment plan. But in the embryology lab, if we have 4 or less embryos on day 3, we like to freeze on day 3 to make sure that you have something to transfer. When we grow the embryos from day 3 to day 5, we expect only about half of them to make it to the blastocyst stage for freezing or transfer. So this is just a natural selection process that happens. So by holding the embryos, it's allowing us to select and freeze and transfer only the ones that are showing that they're growing strongly and are viable. So if you have, 10 embryos on day 3, we'd like to hold those out. And then when you come for your FET, you can just transfer one good embryo instead of transferring some day 3 embryos. If you have so many to choose from, we don't really know which one to choose. So you might end up going through more transfers. But either way works. What does cryo? So if you get a message that says they cryopreserved 11 day 5 and 6 embryos, I think they're meaning they froze 11 total embryos on day 5 and day 6. Rosette, I think that's what they mean. That's a good question about Pgta. Does Pgta normal trump any grade? So if it's normal, it's considered good. I would generally say yes. The Pgt results are more important than the grade. We would choose, a fair to poor normal embryo rather than transferring a good graded abnormal embryo. So once you have those Pgt results, we're obviously going to transfer the normal embryos. And like I said before, sometimes the really good looking embryos end up being abnormal. We can't see the chromosomes under the microscope. We can just see the cells, and we think the cells look good. But really, when you did the Pgta, they might not come back as euploid. Congratulations. Hi, Jill. Okay, so the biopsy process. So embryo biopsy is a really advanced technique that we start to learn once we've been in the lab for about a year. So becoming an embryologist is a lot of hands-on training, and it takes a good 6 months until we're actually working on patient samples and very comfortable and injecting eggs. And I'd say like a year until you're fully trained. So we put in a lot of hours and we work really hard. We practice a lot. That's why if some of you donate your embryos that you're not using anymore, if you donate them to the CNY training program, we thaw those embryos, and we'll even use donated embryos just to practice freezing, thawing, biopsying, moving things around, and then they're discarded safely. So that's what those are used for, if you were ever wondering. So an embryo biopsy, we are trying to take about 5 cells from the trophectoderm. So again, the trophectoderm is that outer, all these cells on the outside. We don't want to take any cells from the inner cell mass because those are already committed to becoming the baby. So embryo biopsy is pretty cool. So we have one pipette that is a holding pipette, which is holding the embryo still. And then we have a suction pipette that we start to kind of pull the cells a little bit. And then we have a laser, and the laser shoots a few punches to help break up those cells. And we kind of like laser and suction the cells, and we pluck just a tiny little bit off. And then we have to load those tiny, 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 tiny cells into a tiny, tiny, tiny little tube that gets sent to Cooper Genomics for the DNA analysis. And the embryos stay at CNY Fertility. They're frozen. So it's just the few cells that we biopsy that we send off to be tested. Sometimes that's confusing. People think we're sending their embryos away and bringing them back from Cooper. That's not true. We're just sending the little biopsied cells to be analyzed. The embryos stay here. So that's a great question, Paige. Can you test embryos after they were already frozen? Yes, we can, and we do this probably every week. It is a little bit risky depending on the quality of your embryos. So our thaw survival rate for embryos is like 99%. We expect the embryos to survive the thaw. But then in order to be biopsied, they have to expand and they have to kind of resume growth and be like have enough cells in order for us to safely take a sample away from them without damaging them. So if you have some good blastocysts frozen, it's pretty likely that we'll be able to thaw and biopsy them. If you have some blasts frozen that have a poor quality trophectoderm, it might be very difficult or impossible for us to take safely from the trophectoderm without damaging the embryos. So it really depends on what you have frozen. But yes, we can do that. It can be a little stressful. So we just want people to know that there are risks. So even though we say we treat every embryo like it's our own, we can't guarantee that they're going to survive that process. Okay, so let's see. I was going to talk about FETs as well, so I see a few questions that relate to that. So when you are doing an FET cycle, so frozen embryo transfer, you're going to talk to your nurse who's going to schedule your monitoring appointments, and then she lets the lab know what day we have to thaw your embryo. So we do all the embryo thaws in the morning. So you want to talk to the nurse about how many embryos you're going to thaw for your transfer. If you're under 35 years old, or if this is your first transfer, we strongly recommend transferring 1 embryo. I know that by the time you get to a frozen embryo transfer, everyone wants success so badly, and we want success for you too, but we also want it to be safe. And if you transfer more than 1 embryo that's good quality and you're young, you have a very high risk of pregnancy with multiples, which is high risk. It's considered high risk for both the babies and the mother. So we follow ASRM guidelines, and we really try to counsel appropriately when it comes to how many embryos to thaw, especially if your embryos are Pgt tested. If you have Pgt euploid embryos, you should be transferring 1 embryo. Okay. So all of our embryos are thawed in the morning, and your ID is doubly checked. So 2 embryologists check the ID of what we're thawing, so we can ensure proper identification. We are very careful. A lot of the steps in the lab are verified by 2 people. And I brought a cryolock. So this is what the embryos are frozen on. So it's a very thin piece of plastic, and it has a top. And where the little black dot is, we put the embryo right above the black dot, and it's plunged into liquid nitrogen. So it's a bit of a process, but this is the little cryolock. Each cryolock will have one embryo, and then we put about 8 cryolocks together in one cane, and that goes into the liquid nitrogen tank. Is the remaining sperm also frozen after you inject all the amount needed into eggs? So no, if you want sperm to be frozen the same day that you have a retrieval, you're going to have to make an appointment with the andrology lab so they know ahead of time that you want to freeze excess sperm. So once they process that sperm for us, we don't want to freeze it once it's already been processed and gone through the gradients. But if ahead of time they know and it's on their schedule to freeze extra sperm, they can split the raw sample in half and process half of it and give it to us and freeze the other half if it's good, if it's a good quality sample. Thank you. If you lose most embryos after a day 3, is that usually a sperm or egg issue? That's a good question. The honest answer is that it takes both. It's kind of suggested out there that if you have a lot of attrition from day 3 to day 5, it's the sperm. And that's because the embryos are using the maternal, they're using so all of the cytoplasm, all the energy they're using is coming from the egg in the first few days. And then when you get to day 3, the embryonic genome is activated. So the egg and sperm are working together to continue development. So if it looks like it's going good until day 3 and then they kind of peter out, sometimes people suggest it's sperm, but it really takes 2 and it's kind of how they go together. So it doesn't necessarily mean that it's sperm. Would you say that if someone's 32, is all eggs that fertilize make it to day 5 blasts? Yeah. If all of your fertilized eggs make blasts, that's an anomaly, to be honest. So an attrition rate is something else that I wanted to mention too. So not all of our eggs that we retrieve are going to be mature, and we can only inject eggs that are mature, so they're ready to be fertilized. So you can retrieve a bunch of eggs, maybe 90% of them are mature, 80, it really varies. And then once we inject the eggs with sperm, we only expect about 75 to 80% to fertilize. So some people have 100% fertilization, some people have 50%. The egg quality and the sperm quality is going to affect those numbers, but we expect about 75 to 80% of the eggs to fertilize. And then most embryos will make it to day 3. And then we lose, like I said before, we expect about half of them not to make it to the blast stage. So there is there's an attrition rate. So at each stage, you might start with 10 eggs and you might only get 3 blasts, which seems like, oh, I should have 10 embryos. And that's just not really how it works. But having 3 embryos from a cycle is amazing. That's also really good. What do we do with non-mature eggs? So we'll check them throughout the day to see if they mature. And if they do mature, we'll inject them that day. So when your partner provides a sample, I don't do this part, but I believe the way that it works is he has to initial next to, we pre-print a label with his name and date of birth and your name and date of birth, and he has to initial that he's witnessing that is him, that's his name and date of birth, and then the person who's giving him the cup also initials, and then the andrology lab, they have double verification too, and then when we get the sample, we're also checking the name and date of birth against the consents. I don't know if I answered that really well, but it does have your name, date of birth, his name, date of birth. And like when we're doing an egg retrieval, if I'm doing a retrieval, when I go to the OR to retrieve the tubes to look for the eggs, I take your sticker with your name, date of birth, his name, date of birth, and it's on my glove. And I wear that sticker the entire time that I'm working on your eggs. So every time I go to the OR to get more eggs, I take another sticker off your door, and that matches up with your card and when we ID'd you. So it's kind of like I'm wearing your name because I'm working on this patient. So I know, and then I'm checking the name against the incubator against the dish. So we keep it consistent. Okay, let me see if I have any other information. Oh, one thing that I wanted to request is we have to report all of our cycle outcomes to the CDC, and we're in the process of wrapping up our data from 2020. So if you had an embryo transfer in 2020 and you had a positive outcome, you have to let us know because we have to report that to the health department. We have a lot of missing pregnancy outcomes right now. So you can send a web message to Embryology, you can send an email, you can call your nurse, whatever way you can get a hold of us. If you are successful, or unfortunately, if you have a miscarriage, we do have to record all of that information. If you did have a baby, we have to know date of birth, weight, and if it was a boy or a girl. So my people who enter data asked me to mention that today because they might be calling you. I waited to use my best embryo for my second transfer. Do they prefer you to use the best embryo first? I mean, it's up to you which embryo you want to transfer. We just generally always recommend transferring your best embryo first. If you have some sort of issue with your preparation for that transfer, or you're uncomfortable with your lining, or whatever you decide with your clinical team, if you want to use one of your fair embryos first, I mean, it's totally up to you. They're your embryos. I work in Syracuse. Thank you. Zymot. So Zymot's kind of new. We are using it. We like it. It's kind of debatable how much of a difference it makes, but it's certainly not going to harm your sperm sample. There is an extra cost for it just because we have to order the microchips. Yeah, no, we don't mix up sperm samples here. It's the number one rule to being an embryologist or an andrologist is attention to detail. And like I said, we have, double ID verification through all the critical steps. All right. So let's see. The best way to get a hold of Embryology is through the patient portal. We're not near the phones very much because we're in the lab working. So if you send a message to whichever lab location, like Embryology Syracuse, Embryology Albany, Colorado, Buffalo, if you send us a web message, we usually get back to you that same day or the next morning. Pgta testing, there is a fee for us for the biopsy fee, and then Cooper Genomics has their own fee. I don't know what that is. I think it's a few $100s per embryo, but I don't want to quote that. Thank you, Lindsay. What is the likelihood an embryo will survive thawing? Our thaw survival rate is about 99%. Our embryos survive really nicely, especially if they were frozen by us. So anytime that you're freezing and thawing embryos or eggs, it is recommended for the best outcome to do it at the same place where you froze the embryo. For Zymot, you have to have at least 1 million total motile sperm. So if it's 8.6 million, you would have enough. You just have to have enough for the sperm to swim through the chip. So Anastasia, I recognize your name. I'll look at your chart when I'm done. Let's see. All right. What do you think? Yeah, that's awesome. I could just go forever, I guess. No, but you can. Thank you for watching. This was fun. If you liked this, let us know. I'd be willing to do this again, and maybe I can have more specific topics to talk to you about. But I think that's a pretty good wrapped up. Oh my gosh, the questions are coming.
I know.
All right. Oh, that's a good thing. When do you get your final report? Once we're finished freezing all the embryos that we can, even if you have a transfer and then we're freezing some, you'll get a report in your portal at the end of the cycle. So the only time it would be delayed is if we finished freezing on a Saturday or Sunday. We upload the reports on Monday, but you'll get pictures, grading information, and how many were frozen. And all of our labs use the same protocol. So if you're transferring between Albany, Buffalo, Colorado, Syracuse, we are all standard. We use the same protocols, so you're fine. I didn't mean to scare you there about transferring your embryos. They'll be fine. Okay.
All right.
All right.
Yay.
Thank you, everyone. Maybe I'll see you later. Thanks.
Amazing. Thank you. That was a lot. And man, I realized just listening that it's a lot more in-depth and complex than I could ever imagine. So thank God we have her.
Ooh, I'm like sweating.
Yeah. Thank you. And, I'm going to try to continue to have people come on. My goal is to have Andrology on as well, and so that they can put the two pieces together. And then, we'll start to get a whole picture of what's going on here. Thank you. Have a wonderful weekend. Happy November. Thank you, Maureen.
You're welcome.
Good day.